Okay guys, welcome. I'm going to try this. So what I'm going to do is at each slide I have parts of the study guide. I'm not going to go over all the answers. However, I will go ahead and talk about some points and give you some pointers for the test too as we go. All right, let's go. So the first thing, we have lipids here. Of course, you need to know what are the two parts that make up the triglycerides, which would be triglycerides, 3 fatty acid, glycerol backbone. 3 examples are your fats, oils, and waxes that cushion, store long-term energy. That brings me to the example. When you do have energy, if you are working out, which energy do you use first? You will use the carbohydrates first, and then after about 20 to 30 minutes of working out, you will start to burn these lipids. There are the other lipids, which are your phospholipids, and your phospholipids are going to make up the cell membrane. Difference between unsaturated and saturated fats we have here would be the double bonds between the unsaturated fats, which would make them a little bit more healthier than the saturated. The test for lipids, the brown paper bag, 3 functions of lipids we've already gone over. Looking at the next screen, you have carbohydrates. What is the function of a carbohydrate? Store energy, monosaccharide. This causes a lot of confusion. There's 2 types of monosaccharides, glucose and fructose. How do you test for polysaccharide? Iodine. Monosaccharide would actually be the Benedict's. What's the color change? Black, brown for polysaccharide, red, orange for monosaccharide. 3 types of polysaccharides. You have your glycogen, starch, and cellulose. We cannot digest cellulose. That's also known as fiber. Glycogens are made by animals. Chemical formula C6H12O6. Draw a glucose molecule. That would be the hexagon. Draw a polysaccharide. A whole bunch of hexagons linked together. So a long distance runner is most likely going to eat before a race. You're going to want to eat carbs with some fat, but the carbs first, because the carbs actually will be burned first, and then, like I said, even longer would be the lipids. Let's see what else with the carbs and lipids that I have here. Just give me a second. If I do a lab where I set up some test tubes and I have a beaker and I do a hot plate and I use Benedict's, please know that that would probably be a carbohydrate test, specifically glucose monosaccharide. What is the importance of carbohydrates in a plant cell? That would be to provide structure, specifically going to be the cell wall. Like I said, what elements make up carbohydrates? That'd be the chemical formula here, C6H12O6. So C would be carbon, H would be hydrogen, and O would be oxygen. So carbon, hydrogen, oxygen would be there. Like I said, please give me a little bit of time because I'm going to go over some of the tests. I'm looking at some of the tests right now. Okay, so let's go to the next page, which would be proteins. Sorry, that's so small. What biomolecule determines which proteins are produced in the body? DNA, deoxyribose nucleic acid. How do enzymes affect the rate? They lower the activation energy, which brings me to this. You do need to know the chart that has the comparison between with an enzyme and without an enzyme. It's in your enzyme notes. The building blocks, amino acids, what bonds them together? Peptide bonds. What do enzymes attach to? A substrate. With this, you need to know a picture of an enzyme substrate complex. First, you have the substrate, then you'll have the enzyme. Then when they bond together at the active site, you're going to produce products. The products don't always have to be 2 separate units. It can actually go in reverse. It can have 2 separate units as a substrate. They come together, and it comes together as one as your product. If I give you an enzyme substrate picture, make sure you can tell all the stuff here. Then we have, like here, draw a picture. What are 2 functions of proteins? You're going to have to speed up chemical reactions or to make structure. The test here is the Biret's. Make sure you know it's blue, and then that it will turn to a violet to black color in the presence of a protein. Another name for a protein, a lot of you missed this on your cutouts, is polypeptides. Let's see what questions I can ask you with that. If we have liver, and we put hydrogen peroxide with it and it bubbles, make sure we know that the liver contains catalase and that the substrate would be water and oxygen. If we heat it, it would actually turn into, it would denature it, so it would not work. Make sure you know that. Along with enzymes, if you have a chart that shows you different enzyme reaction rates, make sure you know that the optimum range of the enzyme functioning would be at the top of the chart. Let's see if I have any other things with enzymes or proteins. Proteins are made up of amino acids. And that's it. Let's go to the next slide. So nucleic acids, what is the function? It stores genetic information. You have two locations, cytoplasm, and then nucleus for DNA, where that is where RNA is made. RNA will also go into the cytoplasm. The monomer is going to be a nucleotide, sugar, phosphate, and base, nitrogen base. Two examples, RNA and DNA. Know that DNA is double-stranded, so that's how, if you're looking at the two, you can tell the difference between a DNA and RNA. DNA is on the back, a picture of DNA is on the back of your notes. DNA determines everything in your body. Let's see. I'm looking at other things. The biggest difference here is that DNA contains deoxyribose sugar, RNA contains ribose. Then we have going into different things with DNA and RNA. Know the structure. If I give you a picture, I've already said that, give you a picture of DNA. Let's go to the next one. General questions. How do you know something's organic? This goes into the organic notes. That would be carbon. It came from something living at one time. Yes, it did, or it had to cycle through. Don't forget you have that funky one, CO2, which is going to be an inorganic compound. What substance is a universal solvent? I like this one. That's water. And in fact, water is going to be part of a solution. So solvent would be the solution. Solute would be what is being dissolved. Let's see, what is a polymer? A polymer is a macromolecule that is made up of tiny units. The number range that makes up the pH scale. Regarding pH, if I talk about an acidic environment, you need to know that we're talking about anything below 7. And also, along the lines of pH, know that extreme acid or base can denature it. Once again, what does denature mean? Please forgive me with all the ums because I'm trying to figure out different questions here for you to help you for the test. Yeah, so what is an acid? If it has more concentrations of hydrogen, you'll know it's an acid. So what is it would be for a base? More hydroxide. We went there. Here's the biggest part for you. Make sure you know diabetes and hemoglobin. 5 to 6 sentences. There's your essay. Compounds organic. What am I looking for here? If it has C, don't forget I have this funky guy, CO2. What is an element? It can't be broken down any further. Going back to this term right here, polymer. So polymers are made of monomers. Monomer is a monosaccharide, glucose, those are example, but also I've been seeing that on a lot of the stuff I was citing and I've talked about. Monomer can also be interchangeable with the word subunit. So if I ask you what is the subunit of something, that's the same thing as what is the monomer. What is extra glucose stored as in plants? That would be starch. In animals, we have it as a glycogen. And actually, that's kind of a weird question. You can see that when animals eat their glucose, it's what we need at the time. If we need more glycogen or more liver, more muscle, we will do that. We will store it as glycogen first and then store it as excess fat. It just depends on where we are with our body metabolism at that point. What are the reactants and products? That's going to go along. Reactants / substrates are going to be the left-hand side. Products are going to be on the right-hand side. So this is going to be a lot having to do with enzymes and their reaction. Another name for an enzyme would be a catalase. Foods that contain carbohydrates, pasta, protein, meats, your good fats will be your butters, things like that. Going into fats and things of that nature, make sure you know that liquid at room temperature would be unsaturated or polyunsaturated. Solid would be butter, things like that, saturated fats. Scientific methods. So as I said, I am going to give you an experiment. It's an experiment that you have not seen before. Be able to tell me what the controls are, what control groups are, what the experimental group is. The difference between controls are things that stay the same throughout the experiment. Control groups, y'all, a lot of you missed this on your quiz. A control group is the side that I did not do anything to. I did not change. I left it as the normal situation. Controls are actually things that stay the same. For example, I left them in the same environment. I used the same type of bowl I use the same instrument, same temperature. Why do we use the scientific method? To explore and learn about the world. So biologists will learn about life, so that is why it's called biology. It's the study of life. A theory is something we come out at the end. It is always, it can be revised or replaced. We look at the world and try to solve problems, so that is why we form a theory. Specifically, after a theory, you have a hypothesis. Two types of data, quantitative, qualitative. Make sure you know qualitative would be a description, like I see that this is occurring, or quantitative would be more of like I counted that there were 7 students who felt better. And characteristics of life, I will give you examples of characteristics of life, and you will need to say which one it pertains to, sort of like on the quiz that you took last week. I hope that is it. 11 minutes. Hopefully I didn't bore you. Go back over this, answer some questions. I'm going to look last time at the quiz, or the test, to see if there's anything. Yes, the structures that are on the back of your colored sheets for biochem, make sure you know those. For example, what does a starch look like? Well, it's squiggly and it has lots of glucose molecules. What is the cutout that I gave you? What does the protein look like? You have the dipeptide bonds and the amino acids. The triglycerol molecule that I have. Anyway, have any questions? Email me. Good luck studying. Hope all is well.